Hi there, welcome back to Bio 23 Infectious Disease. Today we're going to go over Chapter 16, in which we talk about how to control microbial diseases. First, going to start off with disinfection. Disinfection is defined as killing or inactivating microbes that cause disease. They date back to the 18th century, a time when people were ignorant of pathogenic microbes and yet still employed methods that controlled diseases of animals and humans. This included things like physical control methods, chemicals, and biological methods like burial. The first two, physical and chemical methods, remain important means of controlling and preventing infectious diseases today. However, biological means, by burial, preventing microbes from spreading that way, is no longer really used. Some of these infectious diseases are controlled by physical agents, such as changing the temperature. This includes pasteurization. Pasteurization is named after its inventor, Louis Pasteur, who in 1856 first developed a process whereby beer was heated to really hot temperatures for a short period of time in order to kill some microbes within the beer. This heating of liquids for a short period of time just reduces the number of microbes and doesn't kill all of them completely. But it is enough to render the beer safer for human consumption. At the turn of the century, scientists discovered that cows could also spread diseases such as tuberculosis through milk. By 1907, pasteurization of milk was implemented and the number of disease outbreaks associated with milk plummeted. Pasteurization is still considered an extremely important process in order to reduce the rate of infectious disease, both in the United States and worldwide. Chemical agents are also used to control microbial diseases. Heavy metals are very reactive and are especially useful in really low concentrations, making them particularly advantageous because only a small amount is needed in order to regulate microbial control. Heavy metals include things like gold, silver, and copper. Even a few drops of copper sulfate can kill algae in fish aquaria. Recent studies have shown that copper in sinks and surfaces, countertops, and pipes can cut down on the number of bacteria in hospitals by 97%. This can reduce hospital infections by 40%, a quite beneficial advantage. You may have even seen silver being used in clothing or bandages. Lululemon and Under Armour are two clothing brands that have in the past, and possibly even now, incorporated silver threads into their clothing to reduce odor and microbial growth. Both pasteurization and heavy metals affect protein function. Pasteurization destroys proteins by causing them to lose their shape or denature. Heavy metals cause effects on protein function by binding to the proteins themselves and causing them to become inactive. Boiling water works in a very similar way as pasteurization, where the temperature is actually brought up to 100 degrees Celsius. If kept there for a long enough period of time, the water can become sterile. Most often, though, boiling water is only kept at hot temperatures like 100 degrees Celsius for about 5 minutes. Again, this is just to reduce the number of microbes, not eliminate them completely. There are also autoclaves that your book has a picture of, and the autoclaves also use heat and steam combined with pressure to destroy proteins in pathogens and kill them. Autoclaves, because they can get up to temperatures over 100 degrees Celsius, can in fact sterilize equipment. Autoclaves are often used by dental offices, research labs, and even some nail salons. Other means of using heat can include incineration, in which case open flame, such as a Bunsen burner or an incinerator, essentially eliminates all water and completely destroys all molecules in a pathogen. These again are sterilization measures, in that they will kill every microbe on the surface of that object. Then, of course, there are other chemical methods. Your book also mentions disinfectants on inanimate objects and surfaces. We generally don't use disinfectants on the human body because they are highly toxic in many cases. Antiseptics are disinfectants that can be used on body tissues. This would be things like alcohol, rubbing alcohol, hydrogen peroxide, and Purell, which can use rubbing alcohol or ethanol, another type of alcohol, the kind that we drink. In addition, using soap and water can also be highly effective. Soap and water physically removes contaminants and their toxins from surfaces. The goal is not necessarily to kill anything, but instead to physically remove them, which can be advantageous if there's a lot of dirt or biological material that's combined with the bacteria. Purell, hydrogen peroxide, and other chemical methods would not necessarily kill all the microbes because they could be hidden underneath dirt particles or within biological materials such as saliva, blood, or feces. Alcohols generally function by destroying cell membranes, viral envelopes, and also destroying protein structure. Interestingly, hand sanitizer only affects some viruses, but not all, including the common cold viruses, some of which can still remain active even after using hand sanitizer. Hand sanitizer can destroy the flu, however, so it's still very useful in many cases. Hydrogen peroxide can also affect and destroy bacteria, and is particularly useful in killing bacteria found in puncture wounds and scrapes, where anaerobic bacteria can grow. There are also halogens that are really commonly used as chemical control agents. Halogens include chlorine and iodine, in particular in the United States. Chlorine is used by many cities to treat their water sources, including the South Bay in the Bay Area of California. Chlorine is also used to treat water in pools and fountains, and, of course, is also a component of bleach, which you can use on surfaces, fabrics, and so forth. Iodine, another halogen, can also be used to treat water. Iodine is also used not only to treat water, but can also be used to treat skin in the case of burn victims or during blood donations or when syringes are needed. Finally, another component that we see in disinfectants are quats. Quats are disinfectants that are used in surgical soaps. They are also not uncommonly found in restaurants, hospitals, and other locations. They have a similar structure to soap, but work in a slightly different way, in that they're not used primarily for the physical removal of microbes, like hand soap, but rather are used to kill microbes, specifically by creating leaks in the microbial cell membranes. Some household hand soaps used to incorporate antibiotics as part of their functional components. The idea is that the soap would physically remove the microbes from the hands while also simultaneously wash them away and kill them, be it on your hands or in the sink. However, it was discovered a short time later that the antibiotic agents would not function effectively in the short period of time that they would be combined with the microbes on your hands. The microbes would then remain alive, but be subjected to small amounts of antibiotics, meaning the bacteria could then become resistant to the antibiotics over time. Additionally, some of the antibiotic agents had unwanted consequences in the human body, including generation of allergies, disrupting hormones, being toxic, and accumulate in the body, leading to microbial resistance. Washing hands with plain soap and water is the best way to get rid of germs. If soap and water are not available, alcohol-based hand sanitizers are the next best thing. Hand sanitizers are effective at reducing the number of microbes on the skin and killing them in the process, but as mentioned previously, they don't remove dirt, grease, blood, or other biological fluids, all of which can shelter and house microorganisms, allowing them to stay alive even with hand sanitizer use. In the next section, we're going to talk about antibiotics.